Today we are going to talk about some specific examples of acute infection. So we have two lectures this week about different kinds of infections. Today is acute and next time is persistent. So what are the differences? We categorize virus infections in two ways, basically. Acute, which are very short-lived, or not short, but they end at some point, either in death of the host or survival of the host. And examples are shown on this graph. So this graph shows you virus replication or reproduction on the y-axis and time on the x-axis. And the blue line is virus reproduction, and the red bars are episodes of disease. So the top is an example of an acute infection where you have a defined period of virus reproduction, a defined period of disease, and then it ends. In contrast to the other patterns, all the others are basically persistent kinds of infections that do not end. You get them and you have them for your lifetime. And there's several kinds of those, and we'll talk about those on Wednesday. Today I want to focus on acute infections, and you know examples of these. Rhinoviruses, rotaviruses, influenza virus, SARS-CoV-2, of course, which will get its own lecture. So some characteristics of acute infections: rapid onset of viral reproduction, limited but not necessarily short course of disease. So it's not shortness that's the key, it's limited. And sometimes it can be severe, of course. The severity is not part of the acute definition. Even though acute sounds like it would be severe, the acute just means it comes and goes. Large numbers of virus particles, very efficient spread from one to another, and eventually immune clearance in most cases, in some cases, for acute infections, death of the host. A course of a typical infection is shown here. Again, virus reproduction on the x-axis, on the y-axis, duration on the x-axis. So we have the initiation of infection at the origin there, and then there's a period of virus reproduction. And the red line is the amount of virus present. So it starts to increase and reaches a peak, of course, and then you engage innate defenses and eventually adaptive defenses, and eventually the virus, levels of virus reduce, of course, and then you have memory as a consequence. And so there's no time on the X, as you can see, because it's different for just about every virus infection. A very common feature of acute infections are what we call inapparent acute infections. These are successful infections. What do I mean by successful? The virus gets in you, it enters cells, it reproduces, and you shed virus. That's a successful infection. But you don't have any symptoms. Stuff you feel, that is, there's no disease. So you don't feel anything. There's no apparent disease. You don't get rash. You don't get paralysis. But you do make sufficient virus particles to spread in the human population. Now, how do we know this? Well, we do serological surveys, and we see that many people have antibodies to a given virus, but they don't remember ever being infected because they have no symptoms. So examples are poliovirus, where over 90% of infections are inapparent. They have no symptoms associated with them. SARS-CoV-2 is another one. Maybe 20% of infections, depending on the study, maybe slightly more, are inapparent. In other words, no symptoms. We do have signs, right? There are signs of infection. A sign is something that you can measure. You can take blood or some other fluid from these patients and measure virus. You will find virus. You can measure immune responses. You can find antibodies, for example. There are signs. The virus is multiplying. The immune system is engaged, but there are no symptoms of infection. And these are common public health problems, these acute viral infections. They affect millions of people each year. SARS-CoV-2, of course, is unusual. It just started. It's a brand new virus that's entered people, caused a pandemic, but it's going to be with us forever, and it's going to be causing many, many infections every year, as does influenza virus and norovirus, rhinoviruses, and many, many other viruses. And they're difficult problems because by the time you feel ill, so feeling ill is a symptom, right? By the time you feel ill, the infection may be almost over, and it probably has already spread to someone else. And this is certainly the case with SARS-CoV-2, that you have asymptomatic transmission at a high rate. People feel quite fine. They walk around, they transmit to others. And it's sometimes 20% of the time they never get sick. So they're always mixing with other people and spreading the infection. And only when they do get symptoms, if ever, do they stay home perhaps, but not everyone does that. Of course, during a pandemic, we were ordered to stay home, which is a good public health measure before we have vaccines, of course. All right, first question. Which of the following do acute infections and incubation periods have in common? Incubation period until you have symptoms. The virus is not replicating. No symptoms are visible. Immune defenses are engaged. The immune system does not respond. All of the above. Which do acute infections and incubation periods have in common? Let's see how we did. Almost, yeah. I thought this would be 100. It's all of the above. Transmission may occur via respiratory droplets is the only one that someone picked, but everything else is right. Incubation period, fever peaks, coughing and weakness. So it's all good, all of the above. We're going to talk about 5 viruses that cause acute infections. I don't have SARS-CoV-2 here. It also causes an acute infection because that is going to be later in the course. We're going to have a separate lecture about 2 pandemics, HIV/AIDS and COVID-19 in one lecture. Today, influenza virus, poliovirus, measles virus, norovirus, and West Nile. Why did I pick these 5? Because they represent different modes of transmission. Influenza virus and measles virus are respiratory transmitted, poliovirus and norovirus fecal oral, and West Nile by mosquitoes. And there are also different kinds of viruses as well. Let's start with influenza. So there are 3 types of influenza virus. Remember, this is an envelope virus with a segmented negative stranded RNA genome. 3 types. Type is an interesting word. These are different influenza viruses. If you get infected with a type A, you're not going to be protected against B or C. That's one of the differences. And A and B cause a similar disease, a respiratory infection that we'll talk about. C, mostly inapparent or mild upper respiratory tract illness, common cold. That's another name for a common cold. Only influenza A virus causes pandemics. Does anyone know when the last influenza pandemic was, by any chance? 2000 and what? I heard the right number. Yeah, 2009, yes. 2009, the so-called swine flu pandemic. And the hallmark of influenza is antigenic variation, which we'll touch on today, and we're going to return to when we talk about evolution. Very interesting. So the virus is transmitted from person to person by droplets produced by coughing, sneezing, and talking, which we have discussed before. There are droplets of different sizes that go different distances. Probably mostly the large and the intermediate sized droplets. Direct contact with an infected individual. What does that mean? Well, just touching someone in some way, even just shaking hands, and they've just touched their nose, you shake your hands, and then at some point you touch your nose, you introduce virus to your nose. We do that all the time. We're always touching our faces. In fact, you can avoid most infections if you keep your hands away from your face until you can wash them. So if you spend the day out and about, you're in the subway in particular, try to keep the hands away until, I have to scratch my eye now, so I can't keep my hand away from my face, until you can wash your hands. Or you can carry the hand sanitizer with you. Direct contact with individuals or surfaces. There's some evidence that contaminated materials remain infectious for some time and you could acquire infection that way. That's not the case with SARS-CoV-2. Don't think, mostly it's transmitted by droplets. The virus, you inhale it, so you either inhale it or it enters your upper tract, the portal of entry of these respiratory viruses. So it enters the upper tract through your mouth or your nose. and reproduces in the mucosal epithelium of your nasopharynx, this upper part of your tract here, lined with epithelial cells. It has a nice mucus layer to protect you. It has a basement membrane, of course, but in some cases the virus will attach and start to reproduce in those epithelial cells. The basement membrane remains intact. This virus does not get into your circulation. It does not become systemic. In most people, the virus goes down into the trachea. And so sometimes when you have influenza, you can have a burning sensation under your sternum, which is roughly where your trachea is. And some people, it will also go down into the alveoli and cause pneumonia, which is a more serious disease, typically in elderly patients. Now, if you may remember from lecture, no, you don't remember this, but I'll tell you, from lecture number 5, virus entry, the receptors for influenza viruses are sialic acids. They're sugars attached to proteins. And human and avian influenza viruses attach to different sialic acids. Human viruses, mainly alpha-2,6, avian alpha-2,3. Avian influenza viruses like H5N1. Did you hear they just culled all the pheasants and chickens on a farm up in Ithaca because they got infected with avian H5N1? Too bad. You know, there's the little chicks that they give to schools so the kids can, I don't know what they do. They look at them and say, this is science. But they had to kill them all. It's so sad because it's hard. So the wild birds flying over infects these farms. So avian viruses bind alpha-2,3. They don't infect people very often. There have been fewer than 1,000 H5N1 avian influenza virus infections since we started tracking it. About 800 deaths, maybe fewer. And part of the problem is that alpha-2,3 sialic acids are mostly in our lower tract. So here you see the bronchioles all the way down in the lung. They have alpha-2,6 and 3. The bronchus has 2,3. The trachea has 2,3. So the virus has to get an avian influenza virus has to go down pretty deeply into the tract in order to infect us. So it's rare. However, and you know, the alpha-2,6, which is the human targeting sialic acids, the alpha-2,6, which bind human, are all the way up throughout the tract. However, there's one, so people who get avian influenza H5N1 typically sleep with chickens because they farm chickens and they only have one house and the chickens live in the same house and so they're inhaling deeply all night and they get infected. But there's another way humans can get infected. Our eyes here, our cornea, our conjunctival membrane, the nasolacrimal duct, they all have alpha-2,3 sialic acids. And so it has been documented on chicken farms that you can have chicken farmers get infected. It starts in the eye, and then the virus drains through the nasolacrimal duct, right? That takes your tears from the eyes into your nose, and then it can get to the rest of the respiratory tract. So not as hard as we thought it would be, but still a very rare infection in humans. So we are worried that H5N1 is going to cause a pandemic in people. We'll talk about that later. But so far it hasn't. It hasn't even spread extensively in people. Uncomplicated influenza, short incubation period, onset of headache, chills, dry cough, flu-like symptoms, high fevers, myalgias, malaise, anorexias. Your fever peaks within a day, and then it goes down, but then you get more respiratory symptoms. You get a productive cough. What does that mean? You're coughing up mucus, and this can last for 1 to 2 weeks. Does that sound familiar? Yeah. So it's a long infection, and you often have pain substernal pain because the virus and the immune system is destroying cells in your respiratory tract. So that's uncomplicated influenza. We diagnose it in a couple of ways. Influenza-like illness, fever of 100 degrees, cough or sore throat. That's it. If you go to student health and you have a fever of 100, cough or sore throat, they'll say you have flu. Sometimes they will test you. There are rapid lab tests, but they're not so accurate. They may just give you antivirals right away or tell you to suck it up probably and go take some over-the-counter stuff to make you feel better. And of course, if you have no other known cause of illness, or you haven't been tested for any anything else. So there's a rapid antigen test that you could do in a doctor's office. You take a nose swab and you put it on the test. There's also PCR, viral culture, serology, but they take more time, of course, and have to have a specialized lab to do that. But in a flu season, they will just say you have flu without even testing many times. So here are two experiments that show you the relationship between shedding and symptoms. These are actually challenge experiments. We can do challenge experiments with certain strains of influenza virus, especially in people who are vaccinated. What does a challenge experiment mean? You give people virus in their nose, which we usually don't do for most viruses. But this is one case we can take a strain that's mild and we use people who are immunized. So on the left, we have inoculated people. You pipette the virus in their nose. They make you sit back in a chair and they pipette it in your nose, and then sometimes they put a clothespin on your nose so it doesn't drip out for like 10 minutes. It's really similar to the real life situation. That's how you get flu, you know. You walk around with a clothespin. I'm joking because I think it's crazy that you have this clothespin because it's not the way it happens, but that's what they do. And then on the left, we are looking at virus titers. They take nasal washes at different times, the x-axis, and we're looking at virus shedding and then a symptom score. A symptom score is they ask you every day, how much have you blown your nose? How much does your head hurt? All these things, and they add up and make a score. So you can see the viral shedding. It's sort of beginning before symptoms are very accentuated. These are pretty mild symptoms. So here you have that first time point. You have a lot of virus shedding. So you have about a day of shedding before you really feel lousy. And so that could infect other people. And then the shedding peaks at, look at this, 2 days. This is really rapid. And then goes down, and by 8 or 9 days, no more virus. All right, so that's one. And then this is a second challenge study on the right where we're looking at more parameters. Again, on the left, we have antibody. Actually, we have multiple y-axes. We have antibody titer, we have virus titer, and the nasopharyngeal wash. And then on the right Y, we have interferon titer in the nasopharyngeal wash. So you take a little bit of PBS saline, you put it in the nose, and then you measure these things. Anyway, on this right-hand graph, we see virus peaking again, 2 to 3 days. Interferon goes up right away, which is what it's supposed to do. We don't have any symptoms here, but these are all antibody. Look, the antibodies are hardly getting going when the virus is gone at day 6. There's a little bit of antibody, but it's mostly interferon that I think is taking care of this infection. And then you have antibodies rising much later, and then of course you get memory for that. But again, you don't know that from this, but you are shedding before a symptom onset. So we track influenza hugely in the world. We'd like to know where it is, and this is an example of one of the trackings over time. So there are many, many laboratories in many countries that will get samples from people, see if they're influenza, and do some studies on them. So here, for example, are influenza numbers of isolates here on the left and percent positive on the right. And so these are different years. So you can see the seasonality of influenza. First of all, here's 2004, 2005. You can see the numbers start going up. So the line is the total number of isolates. And then the bar graphs are the percent positive. You can see it starts to go up in week 50-ish. That's the end of the year, right? 52 weeks in a year. And then the peak is like week 8. Which is February-ish, and then it starts to go down by week 18. More or less similar every year. So winter disease in the Northeast, this is US. And also these different colored bars are the different strains of influenza virus that can circulate. So we have influenza A and we have influenza B. And you can see the B is in green. It was particularly high in 2007 and 8. And in some years, we have more H3N2, and in other years, we have more H1N1, 2 different subtypes, and they vary according to the year. So that's an example of seasonality. Here's a figure showing the burden of influenza in the US. So this triangle on the left shows you deaths at the top. This is for the years between 2010 and 2023. So between
4,900 and 51,000 deaths, depending on the year. Hospitalizations between 100,000 and 710,000, and illnesses between 9.3 million and 41 million. And on the right is a more granular view of those same numbers by year. So you can see some years very low illnesses and hospitalizations and death, and in other years like 2017, 2018, 51,000 deaths. That's the peak over this time period. So this is why we immunize against influenza. Here are some more ways that CDC tracks influenza in the US. In the top left is the yearly percent positive specimens. These specimens are sent to, or reported to CDC by public health laboratories. This is up to March 16th of this year. And they're tested, and you can see the percent positive. And it increases with movement towards the end of the year. So the peak is occurring around week 50 or so, and then decreases subsequently. So now we're in March, and looks like the season is coming to a close. But also here you can see the different influenza viruses that occur during the season. We have the H3, we have the H1N1, and in green, the B lineage. Another statistic at the bottom left, laboratory confirmed hospitalizations for different years. These are rates per 100,000 population, and you can see in some years, like this green one, which looks like 2020, 21, very low, but that was a pandemic year when everyone was staying home. Here is in light green, 20, 21, 22, a little bit more hospitalization. You can see other years have higher levels of hospitalization. 27, 2018, we've already seen. That was a pretty substantial year for hospitalization. So the amount of laboratory confirmed hospitalizations varies from year to year. Top right, pneumonia and influenza mortality. The two are usually lumped together. Sometimes it's hard to know what is causing the pneumonia. And so for each year, you can see different years here from 2015 to 2019. You have an epidemic threshold and a seasonal baseline. That is what you expect to be the highest and the lowest numbers of influenza and pneumonia deaths. And they peak, of course, in the winter. And then the red line is the actual data. These are the data that are compiled for the year for mortality. And you can see in some years it's around what you would expect, slightly higher perhaps. And in this year, of course, 2017, 2018, very high mortality. And finally, you can get reports per state. This is influenza season, week 40, ending October 2022. And the colors show minimal to very high activity on a state-by-state level. Finally, we have percent outpatient visits for respiratory illnesses, that would include influenza, reported by the Outpatient Influenza-like Illness Surveillance Network. And again, this varies according to the year. And so the years are color-coded here, and you can see the green, again, the 2020-21 season, very little outpatient visits for respiratory illness. And in other years, it's higher. Sometimes it peaks week 48 and then goes down, as you can see for the, looks like the 2022-23 season, that orange one. The blue one, which is 2019-20, we had several peaks of outpatient visits going well into the next year by week 12 and 13. So 12 would be 4 months in, January, February, March, April. And we are, the 23-24 season is the red here, and that has a single peak of outpatient visits around week 52, and then it's been declining ever since. Complications occur. You can get pneumonia. As I say, the virus goes down into the lung and makes a lot of damage. You can get secondary bacterial pneumonia. This means you're starting to recover from influenza, and then suddenly the bacteria invade and cause a serious pneumonia. Muscle pain. You can have heart involvement. Remember, your lung capacity is compromised. Your heart is working harder, and in some people that causes issues. And then something called Reye syndrome, encephalopathy, and liver damage.
etiology of that is unknown. We have over-the-counter stuff you can take to make you feel better. We have antiviral drugs, Tamiflu, Relenza, Flumidine, Xofluza. We'll talk about those when we talk about antivirals. But you have to take those within 1 or 2 days of symptom onset. When you first feel that scratchiness in your throat, you got to go get a prescription. If you wait too long, it's of zero use. But if you take them within a day or two, they will help shorten the course, which is already quite short. Right? And we have vaccines, and we'll talk all about those later. Next question. Which of the following is characteristic of uncomplicated influenza? Transmission may occur via respiratory droplets. Incubation period 1 to 5 days. Fever peaks within 24 hours. Coughing and weakness can last for 2 weeks. All of the above. How do we do? Most of you got all of the above. Very good. Only one person, I think. That's what that 4% is. One person got transmission may occur. That's true, but they're all true. 1 to 5 days, fever peaks in 24 hours, coughing and weakness can last for 2 weeks. Measles is our next virus. It's a paramyxovirus enveloped with a negative sense RNA genome. The envelope, of course, has spike glycoproteins. It has a hemagglutinin neuraminidase spike and a fusion spike. And this is a highly contagious human virus. One of the most contagious human viruses. One infected person can infect, on average, 15 other people. So if I had measles and you were all susceptible, you'd be infected after this lecture. All of you, probably. So there's one viral serotype, and if you get infected or vaccinated, you get lifelong protection against infection. This starts out as a respiratory infection. It's transmitted by respiratory droplets and aerosols. And in fact, this is one of the viruses that's very well transmitted in the smallest of droplets that go very long distances. So that's one of the reasons why this is so contagious. It's present in those tiniest of droplets. There's a long period of shedding before symptoms, and you shed a lot of virus. So those three things together combine to make this a highly contagious infection. So that you are shedding 2 to 3 days before the rash. And the rash is what makes you realize you have measles. Before that, you don't know. Kids go to school and they spread it very effectively. And this is one where there are very few asymptomatic infections. Nearly all infected individuals show some kind of disease. So here's the pathogenesis of measles. So you inhale the virus. It doesn't attach to your respiratory epithelium. Here's the respiratory epithelium, typically in the upper tract. The virus is taken into dendritic cells or macrophages. It doesn't attach to the epithelial cell. There's no receptor for measles on the apical surface of cells. But there are immune cells throughout that epithelium, remember, and those have receptors for the virus. They bind it, the virus will reproduce in them. And of course, those cells, DCs and macrophages, bring the virus to lymph nodes. That's their job. And there that reproduces further in lymphoid cells, and then it gets out into the blood, and you have your primary viremia. The primary viremia, virus in the blood, brings it to multiple organs, including the skin, where it makes the characteristic rash of measles. Now, how does it get to another person? So in the blood, there's plenty of virus in the blood. Of course, your respiratory tract is highly vascularized. So below the epithelium, there are capillaries, and the virus is produced in immune cells. And it turns out, so these orange cells here are immune cells that are producing virus, say in capillaries just below the epithelial surface. Turns out there is a receptor for the virus on the basolateral surface of the epithelium. There's no receptor on the apical surface, but the virus can enter the basolateral surface, reproduce in these epithelial cells, and then be released apically, and now you're in the lumen of the lung or the respiratory tract, and that's how you spread it to another person. So it's kind of unusual that there's no apical receptor. It's taken in by immune cells, and to get out, it goes into a basolateral receptor. And you see, among the organs here is skin and brain. So 1 in 1,000 kids with measles get encephalitis, an inflammation of the brain. It's not good to have virus in your brain. So uncomplicated measles, a high fever, respiratory symptoms, runny nose and cough, conjunctivitis, inflammation of the conjunctiva, and Koplik spots, which are these white spots in the buccal mucosa. So this is the mouth of a kid. Those are teeth, by the way, you see. Got some fillings there, that's a tongue. And this is the side of the cheek here. These white spots are Koplik spots. What's happening there is the virus is replicating in the epithelium and causing the cells to fuse. This is a syncytium-inducing virus, and those fused cells appear as white. And so you could actually take a little swab of those cells, put them on a microscope slide, and look at it, and you would see syncytium, and boom, it's measles. That's what they did in the old days. I don't think doctors look at microscopes anymore. Rash from face to extremities. And you see that child has a rash everywhere. So here's a timeline of the infection. We have the incubation period. So infection starts right day 0. Incubation period where you don't feel anything, but the virus is moving around inside you. You have a primary and secondary viremia. You have a prodrome, which are symptoms before those that are characteristic of infection, and then the rash. So that rash is characteristic of measles. Should be antibody, okay? That's been on there for years. You may say, well, Koplik spots seem like they're specific, but some other viruses can cause that as well. So the prodrome is symptoms that are not specific to the virus. Then, and look at the timeline here. This is a long period. 8 days incubation. You're shedding virus. You're also shedding virus in the prodrome. And then finally you get your rash and you say, oh my gosh, we have measles at day 14. You continue to shed, so hopefully you stay home. And then after about 3 weeks, you go into the recovery period. So it's a very long infection. But there are complications, and this is what makes measles severe. Encephalitis, 1 in 1,000 kids. And I don't have to tell you that virus reproduction in the brain is not good. You have cognitive issues, long-term cognitive issues, potentially long-term motor issues. It's just not good. Bronchitis, pneumonia, ear infections. The fatality is 1 to 2 per 1,000 in a country like the US, but in countries where there's poor nutrition, it's as high as 28%. And then a long-term complication is called SSPE. This is a chronic degenerative neurological disease where 10 years after a child has had measles and has recovered, suddenly begins to lose motor and cognitive function, degenerates neurologically, and inevitably will die. And these, the frequency of these may be 1 in 10,000 cases. So when you see outbreaks of measles, this is one of the big problems. It also causes immunosuppression. As we discussed last time, it reproduces in immune cells, leading cause of death in third world kids, and it erases immune memory. Remember, all of your T and B cell memory is gone after you have measles, so you're susceptible to other infections. So this is not something to play with, right? This is a serious infection, and you need to prevent it by vaccination. Before the vaccination in the US, there used to be 3 to 4 million cases a year. I had measles as a kid. There was no vaccine when I went to school. 400 to 500 deaths, 48,000 hospitalizations, 1,000 kids a year with chronic disability due to encephalitis. In 2000, we introduced the vaccine in the 60s. We stopped endemic transmission in the US by 2000, but unfortunately, the vaccine is MMR, measles, mumps, rubella, so we combined 3 viruses in one vaccine. Unfortunately, in 1998, Andrew Wakefield, a gastroenterologist in the UK, published a report saying that MMR caused autism. And this was a completely wrong and fabricated report. Nevertheless, people listened, and immunization rates decreased in the UK. They decreased here as well. And we have had many outbreaks of measles here in the US. And just to give you some ideas about the numbers, these are on the y-axis, reported cases per 100,000 of measles. Vaccine introduced 1961, goes way down, but there's still cases below that line, as you will see. So in the US, we track measles cases. The top graph are cases per year. So you can see some years, a few dozen cases, other years over hundreds. A big one in 2019 here, largely fueled by an outbreak here in New York City. And these are the states currently that have had measles cases. So 2024, lots of states are having measles outbreaks. And here is the curve of measles cases from the beginning of 23 through February 24. You see we're having increasing numbers of cases. Why is this? It's a backlash against mandatory COVID vaccinations. Many states, particularly conservative states with certain kinds of politicians running them, feel that we shouldn't make people get vaccines. And so they are allowing for religious exemptions to vaccination. So they send their kids to school without getting measles, MMR vaccine, among others. And because measles is highly contagious, you see we're getting cases in many states. This is going to be a big outbreak in the next year. You're going to see every state's going to have measles. There will be thousands of cases. There will be kids with encephalitis and SSPE eventually, because people have this idea that they should decide what their kids should get and not the public health authorities. And I just don't get it. I'm sorry. Kids need to get vaccinated against this. This vaccine is harmless, folks. It doesn't do anything bad to people. So that's where we are. Globally, we have lots of measles. By the way, this is the New York City outbreak in 2018, 2019, mainly in Brooklyn, in a community that is under-vaccinated. They don't believe in vaccination. So large numbers of cases there. And globally, these are the different WHO regions, right? Africa, Americas, European Mediterranean, Eastern Mediterranean, European region, Southeast Asia region, Western Pacific region. The WHO divides the world into those regions, as you can see down here. And these are the outbreaks of measles in each of the regions. So, see, Africa had a lot of measles in 2019. And these are the numbers of cases. Tens of thousands of cases of measles. And in fact, in 2022, the cases rose 18%, and deaths went up 43% globally because of COVID and that limited vaccination. There were 413 cases globally in 2019, and in 2022, 136,000 deaths. And this is the global incidence of measles. So the more red, the more cases. So you can see lots of cases in Africa and different parts of Asia and the Pacific. But here, look at the table. Top 10 countries with measles: Kazakhstan, Azerbaijan, Yemen, India, Iraq, Ethiopia, Kyrgyzstan. I mean, Russia, Pakistan, tens of thousands of cases of measles, completely under-vaccinated in these countries. And many of them, as it says here, put off their measles campaigns due to the pandemic. So this is also a follow-up to that, but still many countries are not vaccinating enough. These are cases that should not occur. We have a vaccine that works and it should really be distributed. Question is, which of the following is a good reason to get measles vaccine? There's a 1 in 1,000 chance of acute post-infection encephalitis. There is a 1 to 2 in 1,000 chance of death. Each infected person spreads measles to 15 others. Immunosuppression can lead to secondary infections, all of the above. All right, how did we do? Yeah, I thought this would be a good candidate for 100%. They're all true. Good. All right, poliomyelitis caused by poliovirus, a small 30 nanometer RNA virus, positive strand genome, no envelope, just an icosahedral capsid. On the left, that's an electron micrograph of the particles. The structure in the middle, which we've discussed before, and the RNA inside is a plus-stranded RNA. Here's a cutaway of the particle showing the very thin shell and then the RNA packed inside of it. The pathogenesis, this is a fecally acquired virus. It's acquired by fecally contaminated material. You ingest the virus, it makes its way through the stomach, amazing, right? Stomach, a low pH, all kinds of enzymes. Then you go into the small intestine with bile salts and different, this virus just is completely resistant to that. It passes through to the intestine, reproduces in the intestines, and then is shed in very high amounts in the feces, and that's how it is transmitted from one person to another. This is an exclusively human infection transmitted by person to person. While it's being shed in the feces, it establishes a viremia. And in about 1% of infected people, the virus destroys enough motor neurons to lead to paralysis. So the virus is in the blood, and we believe that the blood brings it to the muscles, and there the virus can enter the nerve endings in the muscle and then travel by retrograde transport to the spinal cord and reproduce in the neuron, destroy that, and that gives you the paralysis. And probably also the virus can cross blood vessel capillaries in the spinal cord and brain and cause paralysis. But that only happens in 1% of infected individuals. As I said, 90% asymptomatic infections and only 1% paralytic. So a well-adapted human pathogen. You may remember my lab, I told you last time, my lab years ago made a transgenic mouse model for polio. And so this is a section of spinal cord from a transgenic mouse that was inoculated with virus. It developed paralysis. And here we see two things. The green dots, that's an in situ hybridization. You take an RNA and you label it and you hybridize it to the section. You look for viral RNA. So all those cells, and they're big cells, you can tell they're neurons because of their size and their roughly pyramidal shape. Those are neurons full of poliovirus RNA. And then you see these dark cells here. These are some kind of immune cell infiltrating into the infected area. You know, they're trying to clear it. And who knows what the relative contribution of it is of those cells versus virus reproduction to damage of the neurons. Nobody ever did those studies. Nevertheless, if you have enough neurons damaged in an area that innervates the muscle, then the muscle will get paralyzed. And we will talk a little bit more about the disease later when we talk about vaccines. But this is a time course of poliomyelitis, which was made many years ago, because you can see it's hand-drawn. You know, in the old days, we used to draw our figures by hand because we didn't have computers. So this was based on observations made in many patients. So you have here different phases of infection. You have the exposure here and then days after exposure. So let's see. Let's stop at the top. We see temperature. So you see an incubation period about 7 days, then you have 7 days, then you have a spike in temperature. You get a headache, sore throat, nausea, and then temperature goes back down again. While that temperature is spiking, you see virus in the feces, you see virus in throat secretions, and there's virus in the blood. Okay, and then in 1% of individuals, the incubation period continues, and then you have another spike in temperature associated with headache and nausea and paralysis. So in those fraction of people, the virus has invaded the CNS and caused the paralysis. And that corresponds to the virus being in the central nervous system. That's what is shown on this bottom panel here. So this shows you, it sort of looks like this happens with everyone, but it's not. 99% of people do not enter this paralytic phase here. So humans are the only reservoir. It's spread by fecal contamination, as I told you, in warm months. In temperate climates, it is a summer disease. So here in the US, kids would get it in the summertime, and you know, in many cities, parents would keep their kids indoors during the summer, not let them go to pools and so forth to try and avoid getting polio. It was highly feared in the 30s, 40s, and 50s. There is a long-term complication. You could call it long polio. It's called post-polio syndrome. So you recover from paralysis with physical therapy and so forth. And then 30 to 40 years later, you start to lose your muscle function again. And this happens in about 25 to 40% of people who have recovered. It's not an infectious process. There's no virus present. We think it is. The problem is that as you age, you lose neurons normally, but usually you have enough of them, so it doesn't matter. But if you've lost many neurons to polio, then as you lose additional neurons, you get this increased paralysis. And there's still people living with post-polio because the big epidemics of polio were just in the 1950s, really. So in the US, many thousands of cases of polio in the 40s and 50s. Peaking in the 50s, the post-World War II baby boomers fueled this big outbreak. We have an inactivated vaccine introduced in 1955. That brought the numbers of cases down. And then an oral replication competent vaccine introduced in 1961, which resulted in eradication of the disease in the US, at least for some period of time. Although 2 summers ago, we had a case of polio in Rockland County, New York. We'll talk all about that when we talk about the vaccine. Okay, but poliomyelitis is very low globally right now, and whether or not we can get rid of it, we'll talk about in the vaccine lecture. Our next virus causes gastroenteritis, right? Did you know in a 24-hour period, about 200 million people in the world have gastroenteritis? This is vomiting and diarrhea, and the amount of diarrheal water that they pass equals the volume of water passing over Victoria Falls every minute, which is 65 million liters per minute. So that's how many people have diarrhea and are passing water. It is, well, it's a disease that makes you lose a lot of water. And most gastroenteritis is caused by norovirus, a small positive sense RNA virus about the same size as poliovirus. Causes about half of all foodborne outbreaks of gastroenteritis in the US. That's 48 million cases. There's a pie chart showing different causes of gastroenteritis. And you can see 58% norovirus, and then a variety of bacteria, Clostridium staph, Salmonella, Campylobacter, et cetera. So the bacteria really are a minor role, although together they make up a lot of gastroenteritis. So most of it is caused by norovirus. So this is one virus that you can do challenge studies with. In fact, it's very typical in some medical schools to challenge students on a Friday. And since it's very hard to grow the virus in culture, you feed them filtered diarrhea, which you put some flavoring in so it doesn't taste so bad. But it's sterile, right? You pass it through a 0.2 micron filter. And then they get sick on Saturday and Sunday, and you can do measurements. And then Monday, they're well enough to go back to class. And I think they're paid $300 for that. So if you go to medical school, they may ask you to do that. But anyway, from those kinds of studies, we know that 18 particles are enough to initiate gastroenteritis. So that means the amount of virus on the head of a pin would be enough to infect over 1,000 people. So that's actually an important piece of information, because then you can figure out how much virus is in fecal material and see how much it actually you need to transmit to someone else. So we call this a 2-bucket disease, because you're vomiting and you have diarrhea. You need 2 buckets to contain it. This is a real thing. I'm not making it up. It's a 2-bucket disease. Fecal oral spread. The viruses, like polio, pass through your stomach and they reproduce in the intestine. They blunt the villi in the proximal jejunum. These are normal villi on the left. These are in a person infected with norovirus. They reproduce in intestinal epithelial cells. We don't know why these viruses cause vomiting and diarrhea. Nobody has figured that out. So the transmission is fecal oral. Sometimes it's vomitous. So I'll show you in a minute. There are some incidences of transmission on airplanes when people are vomiting into those bags. They make an aerosol, and that can infect the entire plane. Food is a big one, water, environmental contamination, oysters and raspberries. Anything you don't wash that comes in contact with water can be a spread mechanism of norovirus. Food handlers who are shedding. So many food handlers are infected and they shed asymptomatically for long periods of time. They don't wash their hands and they contaminate food. That's why there are signs in the restrooms of restaurants. Employees must wash their hands. That's because of norovirus. We found out that a lot of workers shed norovirus, and so it's for them. It's not for you because you don't work in the restaurant, but you should wash your hands anyway. Incubation period, sudden onset of vomiting, diarrhea, stomach pain, 28 to 60 hours duration, 30% asymptomatic infection. So you can have no symptoms and shedding virus, and you're preparing food at a banquet and you contaminate the food and you get a big outbreak. Virus shedding peak 1 to 3 days, but can persist for up to 2 months in the absence of symptoms. And so again, that's a lot of the food handlers are shedding virus. Immunity is not great. Even after infection, you have a very brief period of immunity
towards that strain that you got, and you can get infected with other strains or later in life. So you can have multiple infections with this. It's a lovely thing to consider. Humans are a main reservoir of the virus, but seems to be able to infect non-human animals as well, and we're slowly beginning to appreciate that there are other animals out there that might transmit it to us. All ages are susceptible. These occur year-round. They peak in cold weather, and they often occur, outbreaks often occur in semi-closed environments. What is that? Nursing homes, hospitals, cruise ships, military schools, as you can see, any place where people are together and can contaminate each other. And so what's the treatment? Just prevent dehydration. We don't have any antivirals. Vaccines are in development, but we don't have them yet. And we have 200,000 deaths a year annually, mainly in children who don't get enough fluids to replenish their fluid loss. So here are some settings and modes of transmission. So settings on the left, the big one is restaurants and catered meals, right? 40% of all outbreaks. Then we have retirement centers, nursing homes, hospital, 25%, daycare centers. And then you got your cruise ships. The cruise ships are a big one. They're actually only 10%, but they get a lot of publicity because it's a big deal when a cruise ship has to come back and everyone has to get off and they have to decontaminate the whole ship. The transmission person to person is the biggest role, 88%, followed by foodborne and waterborne. So here's an article about transmission on a flight. This was an Air New Zealand flight where a sick passenger passed the virus to the crew. So the crew cleaned up. So the passenger vomited, obviously. The crew cleaned it up. The crew got infected, and they went on to different flights, and they infected people on those flights as well. So this is what happens when you have asymptomatic shedding for a good period of time. And cruise ships, as you saw, 10% of outbreaks are on cruise ships, and they're well documented. Here are different cruise ships and the sailing dates. Some of them are confirmed to be norovirus. Others are caused by other viruses or they just haven't figured it out. And, you know, when you have an outbreak on a cruise ship is a special environment, right? You have thousands of people crammed onto this boat, and you can have staff that are shedding. You can have passengers who are shedding. And if you're there for a week, you're going to get infected. Personally, I would never set foot on one of these things. If you think that's a good way to spend a holiday, take your risk, but I would never do this. Not the happiest place on earth. That's what Disney calls its cruise ships. So why are they associated with cruise ships? Well, we track illnesses, and so we find outbreaks very quickly. And the close living quarters, as I've told you, and then new passengers coming in all the time can bring virus in and incubate it there. But of course, it's not just norovirus. So remember, during COVID, several cruise ship outbreaks of COVID happened as well. These are just good places to transmit viruses, I suppose. So how do you prevent getting norovirus? You wash your hands frequently. You rinse your fruits and vegetables because they may have been washed with contaminated water before market. Shellfish, oysters are a good source. I mean, raw oysters are great, but you're taking a risk by eating them. Why is that? Because sewage goes into oceans unfiltered, and the oysters are filter feeders. They take water constantly through them, and the virus particles get in them. So when you eat the oyster, you get lots of virus. Or you could drop the oyster in a shot of vodka. Maybe that would do it too. Clean surfaces well. If you're sick, don't prepare food for others. And mostly don't eat the mince at the restaurant when you go out near the cashier. I don't think there are mints anymore, but there's certainly things wrapped in plastic. I wouldn't touch those because, as I said before, people have done surveys, and these things near the cash register are uniformly contaminated with coliforms, which is a measure of fecal contamination. People come out of the bathroom, they have fecal contamination of their hands. They haven't washed their hands. They touch the mints or the packets, and then you pick it up. People are really bad with hand hygiene. All right, our last virus is West Nile virus. This was first isolated in 1937 from the West Nile district of Uganda. It's nothing to do with Egypt, right? So people think this came from Egypt, and you can see this New Yorker cartoon. We're pretty sure it's West Nile virus because there's a
Pharaoh, but no, it's not Egypt. Uninformed cartoonists, I guess. Was not present in the Western Hemisphere until 1999, where it came to New York City in the summer of New York. And it spread throughout New York and then eventually through the rest of the country and then to South America. So now the whole Western Hemisphere is infected with West Nile virus. And that virus probably came, we don't know how it got here, but the sequence of the New York isolate is exactly the same as an isolate from an Israeli goose. So there are goose farms in Israel, and they found the same virus there. So it infects birds of all sorts. So somehow it made its way here. I think it was probably in a child that had an unusually high viremia, because normally when you get infected, your viremia is very low, and it's not enough for a mosquito to pick up and transmit to another individual. But children tend to have higher levels of viremia, so maybe it was a child who flew in, was infected in Israel, and then flew into New York. Who knows? The virus infects many different birds. It's carried by different kinds of mosquitoes, and it also infects other vertebrates besides birds and us. So this is a virus of primarily birds and small rodents. So that's the natural cycle. Mosquitoes carry the virus among birds. So wild birds tend to be relatively resistant to disease, but they perpetuate the virus. Occasionally a mosquito that has picked up a blood meal from a bird, an infected bird, will infect a human or a horse, and then the humans or the horse get disease, sometimes severe disease, but they're dead-end hosts because they don't have enough viremia for a mosquito to bite them and pick up enough virus to spread to another individual. So just think about it. Every case of West Nile is a spillover from a bird or maybe a rodent into humans. It does not go human to human. It's very interesting. So that, by the way, West Nile virus is a flavivirus, enveloped positive strand RNA virus related to yellow fever virus, dengue virus, and others. So it's transmitted by culisine mosquitoes, 3 to 14-day incubation period. About 20 to 30% of people develop a flu-like illness called West Nile fever, and 80% of people have no symptoms, asymptomatically infected. But in this case, it doesn't matter because they do not transmit virus to anyone else, because the only way you transmit is through a mosquito bite. And as I said, humans do not have sufficient viremia to allow a mosquito to pick up virus and transmit it to something else. About 1 in 150 people develop a neuroinvasive disease. In other words, the virus gets into the central nervous system. You get headache, you have eye issues, muscle weaknesses, cognitive impairment, a polio-like flaccid paralysis, and 10% mortality. It's not anything to joke at, and 50% or more of people have long-term neurological sequelae, cognitive impairment, muscle, motor issues, and so forth. And the pathogenesis is, right, the mosquito picks up virus, let's say from a bird, delivers it to a human. The virus is injected into the bloodstream. It's picked up by dendritic cells and brought to the lymph node. And the virus likes to reproduce in cells in lymph nodes, lymphoid cells, so it reproduces there and establishes a viremia. It can then get into the spleen where there are more lymphoid cells, of course, and it can further reproduce. Some of the early responses to infection include sensing by toll-like receptor 3. So it's an RNA virus. It's sensed by TLR3. That sensing induces cytokines, among which is TNF-alpha, tumor necrosis factor alpha. One of the effects of TNF-alpha is to loosen the blood-brain barrier, the barrier between the capillaries and the tissues of the brain. And so that allows virus to get into the brain. We know this because in mice, if you knock out TLR3 and infect them with West Nile virus, the virus is less likely to get in the brain. So then the virus gets into the brain where it can reproduce in neurons and cause the symptoms that I told you. All these immune cells here throughout this figure, these are a variety of immune cells that are thought to be important for control of infection. So of course it's all the players, B cells and T cells of different sorts, interferon complement, and in the brain, interferon and different kinds of T cells. So here's a graph of, or a table of West Nile in the US from 99 when it first came in through 2019. And you can see it's listed as neuroinvasive, non-neuroinvasive, and total cases. So you get a sense for the fraction of infections that are neuroinvasive. So total cases, well, of course, each bar is total cases. Deaths are in orange here. So the light blue are the neuroinvasive cases, which each year is somewhat different. And then the dark bars are the non-neuroinvasive cases. So you can see a couple of things here. First of all, in some years there are many cases, and in other years there are not so many cases. We don't know why that would be. People have speculated about it, but we're not sure. And there are thousands of cases potentially every year. These are U.S. data, obviously more elsewhere. 2019, the average incidence by state. You can see some states have a greater incidence here. Here in the Northeast, we have a relatively low incidence. It's not zero, but many other states, particularly ones where there are many mosquitoes, have high levels of West Nile disease. And this is a number of cases by by month of the year, just to show you how it is seasonal, because mosquitoes are only feeding at certain times of the year. So the summer months particularly, that's mosquito season, so that's where we see most of the West Nile cases. And the age also controls whether you have neuroinvasive disease or not. So this is incidence of neuroinvasive disease per 100,000 population. You can see it's very low at young ages, and then it increases with increasing age, and it's highest over 70 years of age. So a typical picture of how old age is a very highly associated factor for severe disease. How to prevent it? We don't have a vaccine right now. We may have one at some point. Well, these mosquitoes bite from the evening to the morning. So they're night biters. They're not day biters. So if you're going to be outside, if you're going to sit on your porch or sit out in Central Park during the night, because there are mosquitoes there too, you should take some precautions. You should use repellents. In your homes, you should have screens. In fact, many places don't have screens, and that's a very easy thing to do. If you're going to keep your window open, you should have a screen on it. And really, if you're going to hike in the evening hours, you should wear long sleeve clothing and long pants. Many people don't do that, and they end up getting covered in mosquito bites, but it's a good way to prevent infection. There is a horse vaccine available. We don't have a human vaccine, but there is a horse vaccine because racehorses are very expensive. I guess all horses are expensive, but especially racehorse. You don't want your $100,000 racehorse to die of West Nile, and this can kill horses. So there is a vaccine available for horses, but we don't have one yet for humans. And one last word about viruses in the central nervous system. I've talked today about a couple of viruses that can get into the CNS, poliovirus, measles virus, West Nile virus, they can all get in the CNS. But getting in the CNS is a dead end. Once the virus gets there, it doesn't get out. So it can't spread to another human, in other words. These viruses, polio, measles, West Nile, they spread by other routes, polio, fecal, oral, measles, respiratory, West Nile by mosquitoes. That's how they spread. So the invasion of the CNS is a dead end. So it is not something that is evolutionarily selected for in a virus. To say that the virus evolved to invade the CNS is not correct, because the main selection of viruses is to transmit to another host, and going in the CNS is a dead end. So if you ever hear people talking about CNS invasion as a selected phenotype, it's not right, because it's a dead end for human viruses. Now, for animal viruses, it probably isn't a dead end, because animals can eat each other, they can eat each other's brains, and they can transmit an infection that way. But humans, last I heard, don't do that. Although they did at some time many years ago, right? And they transmitted other diseases in that fashion. Next time, we will talk about the infections that do not get resolved, like these, the persistent infections.